If you look at a giant oak tree in the park, or even just a house plant sitting quietly on your windowsill, it's incredibly tempting to view them as the ultimate perfectly engineered solar panels.
Oh, absolutely. I mean, they literally eat sunlight to build themselves.
Right. It seems like the absolute pinnacle of green energy. But if you actually run the numbers, the reality is, well, it's a little shocking. Plants are actually terrible at solar to biomass conversion.
Yeah, it's a harsh reality check.
We are talking about a thermodynamic efficiency of roughly, what, 1% to 2%?
At best, yeah. I mean, they are absolutely wonderful for maintaining the biosphere and, providing oxygen. But if you look at a leaf purely from a human engineering perspective, it's highly inefficient.
Yeah.
Evolution optimized plants for survival and reproduction, right? Not for maximizing energy yield for us to use.
Right, exactly. And to top it off, basically all they produce with that tiny 1% efficiency is sugars and structural cellulose. So welcome back to the Deep Dive Lab. I'm your host, and today's mission is to explore a scientific frontier that sounds, frankly, completely like science fiction.
And I am your resident expert, and I can confirm it does sound like a movie plot.
It really does.
Yeah.
Here is the strange mystery we are unraveling today for you. How can scientists merge living, breathing bacteria with tiny synthetic electronic materials to create microscopic cyborgs that eat sunlight and breathe out rocket fuel, biodegradable plastics, or fertilizer?
I know, it sounds like the origin story of a comic book superhero, but it is very real, and it's actively solving a massive, decades-long problem we've faced in the quest to, move away from fossil fuels.
Which is huge.
Exactly. It all revolves around finding a way to make two fundamentally different worlds, the biological and the electronic, actually talk to each other.
Okay, I want to get to the microscopic cyborgs, obviously, but I feel like we need to understand why scientists went down this road in the first place. Wait, if plants are stuck at a 1% efficiency, why aren't we just building better, purely artificial solar panels?
Well,
because I see solar farms everywhere now. Haven't we already solved the whole light harvesting problem?
we have definitely made massive strides there. That was kind of the original dream of purely artificial photosynthesis. And, to give credit where it's due, human engineering is incredibly good at the light harvesting side of the equation.
Right.
Purely artificial systems made of synthetic semiconductor materials like silicon or perovskites, they're staggering in their efficiency. Some tandem solar cell electrolysis systems can hit over 30% efficiency.
30%. Wow.
Yeah, for converting solar energy into something like hydrogen.
30% compared to the plant's 1%. I mean, that sounds like a total victory. Why are we even bothering with bacteria if we could just build machines that are 30 times better at capturing the sun?
Because capturing the energy is really only half the battle. Here is the massive limitation that engineers ran into. Artificial systems are notoriously bad at complex chemistry.
Bad at chemistry. How so?
They are fantastic at brute force energy harvesting, but they mostly just use that energy to split water into hydrogen and oxygen. Or maybe they reduce carbon dioxide into very, very simple molecules like carbon monoxide or formic acid.
Okay, I think I see where this is going.
Right. They completely lack the finesse required to take simple building blocks and stitch them together into complex, high-value, multi-carbon chemicals.
So basically, instead of trying to teach a solar panel how to be a sophisticated chemist, we hit a wall. Let me see if I can try an analogy here.
Go for it.
It's like having a massive state-of-the-art nuclear power plant. It generates a staggering, mind-blowing amount of electricity. But the only appliance it is actually hooked up to is a basic water boiler.
A water boiler? I love that.
Yeah, it just boils water all day with all that immense power.
That is a highly accurate way to visualize it. Just raw, unrefined power with absolutely no biological finesse. But on the flip side of that coin, you have biology.
Right, the bacteria.
Exactly. Living microbes are absolute master chemists. Over billions of years, they've evolved these intricate metabolic networks. They possess enzymes that can take simple molecules from their environment and effortlessly assemble them into incredibly complex, targeted chemicals.
Which is exactly what the solar panels can't do.
Precisely. And unlike an artificial machine that degrades and breaks down over time, a living microbe self-repairs. And it multiplies.
But there is a catch, isn't there? The specific bacteria that are best at this high-level complex chemistry, the ones we actually want to use, they're generally non-photosynthetic.
They are.
They're completely blind to sunlight. So following the analogy, it's like having a Michelin star master chef who can cook anything, but they're sitting in a kitchen with absolutely no electricity to run their stove.
Yes, that is exactly it. You have the synthetic power plant with no chef and the biological chef with no power. For a long time, these were two completely isolated scientific disciplines.
Just totally separate worlds.
Right. Material scientists worked on the rigid, inorganic solar panels, and biologists and microbiologists worked on the soft, living cells. So the ultimate scientific mystery became a question of translation.
How to get them to talk.
Exactly. How do you plug the synthetic power plant directly into the biological chef's kitchen without, electrocuting the chef?
Which brings us to the actual breakthrough. So how did scientists figure this out? We are looking at a really comprehensive study published in the prestigious journal Chemical Reviews, right?
AR, yeah.
It's by a brilliant team of researchers, including Dr. Wento Song, Dr. Erwin Reisner, Dr. Bin Liu, and their colleagues from the National University of Singapore and the University of Cambridge.
Yeah,
They essentially mapped out exactly how this translation is being achieved.
they did. They are engineering what the field calls microbe semiconductor biohybrids.
Microbe semiconductor biohybrids. That is quite a mouthful.
It is, but it's very descriptive. They are literally fusing the two halves together. The research details how scientists are taking light-harvesting semiconductor materials and physically attaching them to living bacterial cells.
Okay, I have to stop you there because my brain is just picturing a tiny bacterium wearing a tiny, rigid backpack made of solar panels. How do you actually attach a piece of synthetic electronics to a living, squishy, microscopic cell?
Honestly, the backpack image isn't too far off conceptually. The paper details two primary methods for assembling these biohybrids. The first is called ex situ assembly.
Ex situ, okay, meaning outside.
Right. This is the more straightforward approach. Scientists manufacture synthetic semiconductor nanoparticles, the tiny solar panels in a laboratory environment using traditional chemistry. Once they have these synthetic crystals, they just introduce them into the liquid culture where the bacteria are happily swimming around.
So it's like dropping tiny batteries into a swimming pool full of bugs.
Yeah.
Do they just, like, bump into each other and stick?
Essentially, yes, through simple electrostatic interactions. Because the bacterial cell wall naturally carries a negative charge, scientists can engineer the nanoparticles to have a positive charge.
Oh, opposites attract. That's clever.
Exactly. They naturally attract and bind to the outside of the cell. But it's the second method, the in situ assembly, that is genuinely a biological marvel.
This was the part of the research that completely blew my mind. In situ, meaning in the original place. What are the bacteria actually doing here?
Well, instead of manufacturing the solar panels in a separate lab, the scientists simply provide the bacteria with the raw elemental building blocks.
Like what kind of blocks?
Specifically, heavy metal ions like cadmium or zinc, and a source of sulfur. Now, heavy metal ions floating around in the environment are usually highly toxic to bacteria.
Right, I imagine cadmium is basically poison to a bug.
Exactly. So, as a natural defense mechanism, the bacteria deploy enzymes to chemically reduce those toxic ions. They transform them from a dangerous, dissolved state into solid, stable, non-toxic nanoparticles right on their own cell surface.
Wait. So the bacteria encounter a toxic environment,
Mhm.
and to protect themselves, they chemically neutralize the threat. But in doing so, they inadvertently manufacture perfectly functional semiconductor crystals all over their own outer membrane.
That is exactly what happened.
They're quite literally building their own custom-fit solar armor just to survive.
I know, it's incredible. They precipitate the material biologically, and because the bacteria are synthesizing the nanoparticles themselves, using their own membrane proteins as nucleation sites, the physical contact between the living cell and the inorganic semiconductor is incredibly intimate.
Wow.
The integration is seamless, and that intimate contact is absolutely crucial for the biohybrid to actually function.
Right, because having a shiny solar backpack is completely useless if you can't plug it into the bug's internal engine. Which brings up the biggest question, I think.
The interface.
Yeah. How does the power actually get from that solid, inorganic rock on the outside across the biological cell wall and into the living chemistry of the microbe?
This is the crux of the entire biohybrid field. It's a process known as interfacial electron transfer. For this semibiological photosynthesis to work, a photon of sunlight hits the semiconductor armor.
Okay, photon hits the armor.
Right. This excites an electron in the crystal, giving it a huge surge of energy. That high-energy electron must then leave the synthetic material, cross the physical gap of the cell membrane, and enter the microbe's internal metabolic pathways.
It sounds so simple when you say it, but crossing a biological membrane with raw electricity sounds tough.
It is. The scientific term for this is extracellular electron transfer, or ET.
So it's kind of like having to find the exact right biological USB cable to connect a piece of hardware to a living organism.
That's a really great way to conceptualize it. And the Chemical Reviews paper details the literal biological hardware that nature has provided to make this connection possible. The first mechanism relies on structures called multi-heme cytochromes.
Okay, let's break that down for the listener. What exactly is a cytochrome?
A cytochrome is a type of protein that contains a heme group. A heme group is just a complex molecule with an iron atom right at its center.
Iron, like in our blood.
Exactly the same concept. Because iron is a transition metal, it is fantastic at holding onto an electron and then passing it along. In some remarkable microbes, like a species called Schoenella oneidensis, these cytochromes actually stack together through the cell membrane.
They stack together.
Yeah, they link up iron center to form literal biological nanowires.
Wait, nature evolved actual electrical wires.
It did. These protein nanowires can stretch for micrometers outside the cell body into the surrounding environment. They act as a conductive conduit, reaching out, grabbing the excited electron from the semiconductor, and passing it down the iron chain directly into the cell's metabolic engine.
That is just wild.
And it happens at staggering speeds, sometimes up to 1 billion electrons per second.
1 billion per second. That is just incredible. But what if a bacterium doesn't have those physical hardwired cytochromes? Like not all microbes grow electrical wires, right?
Correct. And that is where a second mechanism comes in. Many microbes secrete specialized organic molecules called flavins. You can think of flavins as a fleet of tiny molecular fairies.
Molecular fairies, okay.
These flavin molecules are released by the cell. They literally swim across the fluid gap over to the semiconductor nanoparticle, physically pick up an excited electron, and then ferry it back across the cell membrane to drop it off inside.
So we have hardwired iron chains acting as conductive cables, or a fleet of little chemical shuttles ferrying the power in. That makes perfect mechanical sense. But as I was reading through the research, there was one detail that genuinely made me stop in my tracks.
Oh, I bet I know which part.
Because everything we just discussed involves the solar panels being stuck on the outside of the cell.
Yeah.
But they found a way to bypass the wall completely.
Ah, yes. You are thinking of the intracellular quantum dots.
Yes. Can you explain to everyone what a quantum dot is and how on Earth scientists get them inside a living bug?
Certainly. A quantum dot is a semiconductor crystal that is unimaginably small, often just a few nanometers across. Because they are so tiny, their electronic properties are governed by the rules of quantum mechanics.
Right, the really weird physics.
Exactly. This means scientists can tune them to absorb very specific wavelengths of light and eject electrons with the perfect amount of energy needed by a biological enzyme. And in some groundbreaking experiments with bacteria like E. coli or a soil bacterium called Azotobacter vinelandii,
E. coli, like the stuff that causes food poisoning.
the very same, well, a safe laboratory strain. But yes, scientists figured out how to engineer these cells to actually ingest these tiny crystals.
So they literally feed the solar panels to the bacteria.
Yes. They place semiconductor materials like indium phosphide or cadmium selenide quantum dots directly inside the cytoplasm. That's the jelly-like interior of the cell.
That is a brilliant pivot.
It really is. If the biggest bottleneck in this whole process is moving the electron from the outside environment across the protective cell wall to the inside machinery, why not just bypass the wall entirely?
Right, just put the power source inside the factory.
Exactly. By putting the light-harvesting crystals directly next to the internal enzymes, you drastically shorten the distance the electron has to travel. You effectively turn the bacterium into a self-contained internal solar reactor.
We are literally turning microbes into self-contained, light-powered chemical factories. It's mind-blowing. But let's zoom out from the microscopic world for a minute. If you're listening to this and wondering how a biohybrid cyborg in a petri dish actually impacts your life, we really need to look at the massive stakes here.
The real-world applications are staggering.
Exactly. Why is this specific technology so critical for our future?
Because it has the potential to completely rewrite the global industrial economy. If you look around you right now, almost everything you interact with relies entirely on the petrochemical industry.
Plastics, fuel, all of it.
Right. The fuel that powers transportation, the plastics in our devices, the fertilizers that grow the food on your table. We pump ancient fossilized carbon out of the earth, burn massive amounts of thermal energy to refine it, and release millions of tons of carbon dioxide into the atmosphere.
Just to manufacture the basic chemical building blocks of modern society.
Exactly. It is a linear, heavily polluting process.
We take it out, we burn it, we pollute. But these biohybrids offer a circular, sustainable alternative. Let's walk through a few of the specific real-world examples from the paper. Start with hydrogen fuel.
Hydrogen is a fantastic clean fuel because when you burn it, the only byproduct is water. By hooking up light-harvesting semiconductors to bacteria that contain an enzyme called hydrogenase, we can drive a totally green reaction.
So how does that work practically?
You can place these biohybrids in municipal wastewater, expose them to sunlight, and they will use the solar energy to strip electrons from the organic waste, churning out pure hydrogen gas.
Wait, really? So they are actively cleaning up human waste and generating a zero-emission fuel at the exact same time, using nothing but sunlight?
Yes. It solves two problems at once.
That is incredible.
Mhm.
Okay, what about plastics? You mentioned biodegradable plastics.
That is another huge application. Carbon dioxide reduction. Instead of pumping oil to make plastic, we can take CO2, the primary greenhouse gas driving climate change, and feed it to these cyborg bacteria.
Feed them pollution.
Right. The sunlight powers their internal metabolism to stitch those carbon atoms together into biopolymers. Specifically, they produce something called polyhydroxybutyrate, or PHB.
Let's unpack PHB for a second. That's essentially a natural plastic, right?
Exactly. PHB is a biological polyester. Some bacteria naturally synthesize it and store it as an energy reserve. It's very similar to how humans store fat.
Oh, that's a great comparison.
And it functions and feels a lot like the synthetic plastics we use every day, but because it is biologically synthesized, it is 100% biodegradable in the environment.
We are talking about utilizing sunlight to literally pull carbon pollution out of the air and turn it into sustainable, biodegradable materials. They eat sunlight and greenhouse gases, and they sweat out biodegradable plastic.
When you phrase it like that, it sounds wild, but it's scientifically accurate.
It's almost hard to believe. But for me, the most impactful example in the research involves agriculture and fertilizer. We have 8 billion people on this planet, and we rely on synthetic nitrogen fertilizer to grow the food to feed everyone.
And the way we currently make that fertilizer is, frankly, an environmental nightmare. It relies on the Haber-Bosch process.
Let's break down why Haber-Bosch is such a problem. The air we breathe is, 78% nitrogen gas? But it's locked up in a triple chemical bond.
Yes. 2 nitrogen atoms are bonded together so tightly that plants simply can't break them apart to use them.
Right. So to break that incredibly tough triple bond and convert nitrogen gas into ammonia for fertilizer, the Haber-Bosch process uses brute industrial force.
Massive force. It requires immense steel reactors operating at extreme pressures up to 200 times normal atmospheric pressure.
200 times.
Yeah.
And the heat, too, right?
Yes, scorching temperatures around 500 degrees Celsius. Maintaining those extreme conditions requires so much energy that the Haber-Bosch process alone consumes about 2% of the entire world's energy supply.
That is a staggering statistic. 2% of global energy just to break one chemical bond.
Yeah.
I'm guessing it emits massive amounts of CO2 as well.
You'd be guessing correctly. It's a colossal energy hog.
But the biohybrids offer a workaround.
Yes, through biological finesse, like we talked about earlier. Certain specialized bacteria naturally possess an enzyme called nitrogenase. Nitrogenase can break that triple bond and fix nitrogen at standard room temperature and normal pressure.
Oh, wow. No massive steel reactors required.
None at all. However, the enzyme requires a massive amount of cellular energy to run. So by pairing these specific bacteria with light-harvesting semiconductors, we can feed the nitrogenase enzyme an endless stream of high-energy electrons directly from the sun.
We can convert atmospheric nitrogen directly into liquid ammonia fertilizer.
Entirely bypassing the fossil fuel-driven Haber-Bosch process.
Okay, I love the optimism, and the mechanism makes perfect sense. But I have to push back here, because there seems to be a glaring issue.
Whoa, what's that?
We are talking about dropping highly efficient semiconductor materials, many of which contain heavy metals like cadmium, directly into or onto living organisms. Cadmium is highly toxic. It's not exactly a health food. Doesn't this just kill the bacteria?
That is a highly perceptive challenge, and you've hit on exactly what researchers consider the biggest hurdle in the field right now. It's called the biocompatibility challenge.
So it is a problem.
You are absolutely right. Many of the most efficient light harvesting crystals are toxic, and the problem actually goes much deeper than just heavy metal poisoning.
What else is happening to the cell?
Well, when a semiconductor absorbs strong light, the energy transfer isn't always perfect. Sometimes an excited electron goes rogue and reacts with oxygen in the water, creating what are called reactive oxygen species, or ROS.
Reactive oxygen species. What do those do?
These are highly unstable, aggressive molecules. You can think of them as microscopic chemical wrecking balls. If ROS build up around the biohybrid, they cause severe photodegradation. They will literally oxidize and shred the bacterial cell membrane apart.
Yikes. So the protective solar backpack can actually catch fire and burn the chef alive, basically.
That's a bit dramatic, but chemically, yeah.
How do scientists get around that? I mean, if the materials are inherently toxic or destructive, doesn't that kill the whole concept?
It doesn't kill the concept, it just defines the next frontier of the engineering. Researchers call this the biotic-abiotic interface. You can't just throw a good solar panel at a good bacterium and hope they get along.
Right, they need couples therapy.
Exactly. You have to actively engineer the exact molecular boundary where they touch.
So how do you engineer a boundary to stop a chemical wrecking ball?
On the materials side, scientists are designing specialized conductive wrappers. For instance, they might coat the toxic semiconductor in a biocompatible polymer, or even encapsulate it inside a lipid membrane that mimics the cell's own biology.
Oh, like a disguise.
Right. The shell allows electrons to pass through, but traps the toxic heavy metal safely inside. Researchers are also engineering single atom bridges.
Single atom bridges.
Yeah, these are precise chemical tethers that physically link the semiconductor directly to the biological enzyme. This ensures the electron transfers instantly, almost like a dedicated bullet train, before it ever has a chance to interact with oxygen and form those destructive reactive oxygen species.
So they are building protective cases and dedicated express lanes for the power. What about the bacteria themselves? Can they be upgraded too?
Absolutely. On the biological side, researchers are utilizing genetic modification. They are editing the microbe's DNA to upregulate its natural defenses against ROS.
Giving them better shields.
Exactly. Or they'll artificially increase the sheer number of those cytochrome wire proteins on the cell surface, ensuring the power flows inward much more efficiently. It really requires a holistic, simultaneous co-design of both the living and the non-living components.
It really is an unprecedented marriage of two completely different scientific worlds. Okay, we have covered a massive amount of ground today, peeling back the layers of this mystery. Let's distill this deep dive down to 3 clear takeaways for everyone listening.
Sounds good.
First, while purely artificial solar panels are incredible at harvesting light, they lack the enzymatic finesse to perform complex chemistry. They are the power plant with no chef.
Second, microbe semiconductor biohybrids solve this by combining the best of both worlds. By successfully interfacing light-harvesting electronic materials with living, metabolizing bacteria, we achieve the raw solar efficiency of the synthetic and the master chemist precision of the biological.
Right. And third, the absolute key to making these microscopic cyborgs function is mastering the interface, the electron transfer.
The biotic-abiotic interface.
Yes. Whether the bug is growing its own solar armor, utilizing iron-rich protein wires, sending out molecular fairies, or even ingesting quantum dots to become an internal reactor, bridging that gap opens the door to producing sustainable fuels, biodegradable plastics, and fertilizers purely from sunlight and waste.
And what is truly profound about this research is that it represents a philosophical shift in human engineering.
How so?
For decades, our goal with green technology was simply to mimic nature. I mean, artificial photosynthesis was really just an attempt to copy a leaf. But with these biohybrids, we are no longer just copying nature.
We're teaming up with it.
Exactly. We are actively augmenting life, pushing it beyond its own evolutionary limits to create a completely circular, sustainable chemical economy.
We are essentially giving nature a hardware upgrade to help fix the mess we made. Which leaves me with a final, somewhat provocative thought for you to mull over as we wrap up today.
Oh, I love these.
If this discovery is just the beginning, if we can successfully merge inorganic semiconductors with living bacteria to run microscopic chemical factories, what other living systems could we augment with synthetic materials next?
That's a great question.
Could we design biohybrid plants that glow in the dark to replace city streetlights? Or what about engineering biohybrid cells inside our own bodies, like cyborg white blood cells that use light to manufacture medicine exactly where a disease is detected.
It's a fascinating thought experiment. The boundary between biological life and synthetic technology is blurring much faster than we ever anticipated, and the applications are really only limited by our imagination.
And that thrill of discovery is exactly why we do this. Thank you, as always, for bringing your curiosity and joining us here in the Deep Dive Lab. If your mind is buzzing as much as ours is, be sure to follow and subscribe for more fascinating science stories. We will see you on the next Deep Dive.